not done grading that, and somebody came up to me after. Does this sound good? I'm going to follow here. And said, can you explain this question to me? And I think I made a mistake in one of the questions, so I'll correct that answer. This is a question that says, which proteins or which organelles use post-translational translocation? And the answers were, sorry? So the ER and the Golgi use co-translational translocation, right? Not post-translational translocation. There were two organelles there, the nucleus and the, what was the other one? Mitochondria maybe? So they would both be using the post-translational, right? And I forced you to pick one of the two. So that's, I think I'll remove that question or I'll give credit to everybody. I don't know which one. Makes a big difference. So that was my bad. I probably intended to write co-translational and I wrote post-translational. Alright. Let's get down to business for today. So we've talked about transport of proteins in a couple of different lectures, right? We talked about the transport that takes place as they are being made in the cell. And after they're being made, so the nuclear transport was one of those things where the protein is made in the cytoplasm, it folds up, and then there's a nuclear localization signal that allows it to go into the nucleus. Now there's nuclear transport can occur using those nuclear localization signals, but if you're small enough, the nuclear pores are big enough that you can actually also diffuse in. So not everything that ends up in the nucleus gets there through some active process. So today we'll talk about the structure and function of nuclear targeting signals. We'll see an example of one, and we'll see what happens if you just mutate a single amino acid in that one. We'll look at nuclear hormone receptors that obviously have to go into the nucleus to have their function. This is related to hydrophobic versus hydrophilic signaling molecules. And then we'll also look at signaling that occurs there through direct nuclear signaling or signaling pathways. So I asked you guys to read a little bit, and somebody brought to my attention that I didn't post the lectures, although I did post a pre-class reading, so I hope people had a chance to look at the pre-class reading. And then we'll go ahead and do our first clicker question of today. The nucleus is separated from the cytoplasm by two membranes. This nuclear membrane consists of a water impermeable phospholipid bilayer and various associated proteins. What do we call this? Good. I see some talking to neighbors going on. So yeah, we're not talking about the whole, right? We're talking about the lipid bilayer. Nice job. So this is the nuclear envelope. The nuclear pore complex is the holes in the nuclear envelope. And the nucleus is the whole thing, including the DNA. And nuclear bilayer, I think, is a fun name, but not what we call this. So I'll move this up. All right. So nuclear transport transport is limited to proteins in the cytosol, because remember, once you get yourself into the ER, that's where you're going to go. You're going to go from the ER to the Golgi, maybe out to the membrane, maybe out to the lysosomes, but never back to the nucleus. So the nuclear transport, you can shuttle between cytoplasm and the nucleus, but those are your options. And so here we have that the shape of the nucleus. So the DNA would be sitting right here in the center, and then it's surrounded by this lipid bilayer structure here, which on the inside is coated. The nuclear lamina, it's coated with proteins that we call lamins. And that is really what is connecting to the inside here where the DNA sits. And then the outside of the membrane and this space in between, the perinuclear space, is really continuous here with the ER, right? And we need that because the nucleus holds our DNA. This is where we have the information we need to make our proteins encoded in the DNA, and then the messages will go to the cytoplasm, and then proteins are going to be made here on the ER. So let's see, what else can we see there? Yeah, we have those nuclear pores. They were drawn pretty big, and they are a pretty big structure. So this means that proteins that are small enough can actually just go through. But if you're pretty big, you need to have a nuclear localization signal to actually get in through an energy costing mechanism. But if you're small enough, you can come in. If you're some ions, you can come in. ATP can come in. That doesn't need to be specifically transported into the nucleus. Let's see, is there anything else? So the nuclear envelope was that double layered structure, right? So the inner and the outer membranes together make the nuclear envelope. All right. Then knowing that, the nuclear membrane is continuous with what structure? There's a clear favorite and then a less favorite. Talk to your neighbors and tell them why you chose one over the other. Well, that was very effective. So, the answer is indeed the rough endoplasmic reticulum. There. And this is, what's the difference between rough and smooth endoplasmic reticulum? What is it? Yes?
Rough ER is coated with ribosomes along the membrane.
Right.
In the ER.
So the rough endoplasmic reticulum has those ribosomes sitting on it. They are making protein. So why is that close to the nucleus? Because that's where the messages are coming from, right? So that's the most efficient way of doing things. The messages come out of the nucleus through those pores. They're transported out. Ribosomes find them, sit down, and then the protein is coming out and is what drags it to the ER. That's right there. And so that makes them the rough endoplasmic reticulum. The smooth endoplasmic reticulum is basically the same stuff, except there's no ribosome sitting on there, and we see that sometimes become very specialized, like in muscle cells. It's then used as a calcium storage site, not so much for making proteins. And so this beautiful alien structure is our nuclear pore complex. And you can see that it must be built up of very many different proteins. In fact, we use 30 different kinds of proteins to build this structure. And so what we're looking at here is the cytosolic area, and then here is what sits inside the nucleus. And we see those cytosolic fibrils. They're obviously called that because they're long thready things, so fibrils, and then they sit on the cytosolic side. And these are important for helping to import proteins. And then we have this big ring structure here made up of 2 layers at least of proteins, the membrane ring proteins that are making, excuse me, that are helping to build this ring structure. Then there's scaffolding proteins that really line this whole membrane pocket so that proteins can come through. And then here, these 2 are proteins, this disordered region of nucleoporins. These are proteins that are kind of just sitting there and preventing big stuff from going through just like that. And then down in the bottom we have the nuclear basket, whose function I'm not entirely clear on, but I imagine that this is part of preventing large things from coming in. So it's kind of like a sieve structure to me. So all of this is working together to get proteins either specifically into the nucleus from the cytoplasm, but there's also proteins that are being transported right out of the nucleus. So how many of these pores do you think we have in a nucleus? Random number.
300?
300, 700? More. But those are good numbers. We can have like 2,000 or so of these guys in a single nuclear membrane. And so if you look at this picture, this is an electron microscope picture of a piece of the nuclear membrane here. And each of these little structures here is one of those nuclear pores. Right, this is if you're looking on the face of the nucleus, looking from above, so now we're seeing the circular structure. And here we've sliced through it. So here we have the pore, and then that basket up there, and then here those cytosolic fibrils sticking out. And the dark area in the center are those proteins that are sitting there preventing things from coming in. Well, excuse me. So why do we have so many? Why are so many holes in the nucleus? What do you think comes in or out? Well, what comes out? What comes out of the nucleus? mRNAs, right? Messages are coming out. So they need to be transported out into the cytoplasm so that they can find their ribosomes. So the number of nuclear pores is not the same in all cells, and it can depend on different things. So what do you think it depends on then, doing that? So the size of the cell doesn't really matter. So the nucleus can be part of an active cell, of a very dormant cell, right, not doing all that much, or making tons of protein, maybe it's an endocrine cell. So it really depends on the transcriptional activity of the cell, because those pores are there to transport messages out, so if there's not that many messages going out, we don't have that many pores on the cell. The DNA content isn't really relevant. It really is a matter of how much of that DNA do you use. So we have, what, I don't know, 20, 30, 40,000 genes in our genome, and there's a lot of space in between, and there's species that have a much more compact genome, but they would be using the same genes for the same type of cells. So the DNA content per se doesn't really make a difference. It's really about what part of the DNA is getting used. All right, so we talked about the amino acid sequences that become this nuclear localization signal. And here we see a picture of what cells look like if they have a fluorescent protein with a nuclear localization signal. That can look like this: lysine, 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 arginine, lysine. And if you mutate a single amino acid, this thing is localized differently. Now, just looking at this for the first time, you might think, well, aren't they all just cells and cells? So what we're looking at is a cell with some shape, and then there's a nucleus inside it. And the protein is the only thing we see, right? The fluorescent protein is the only thing that we see. And therefore here, what you're seeing is only the nuclei. There's a cell sitting right around it that we don't really see. So that's why with the nuclear localization signal, you see the cell light up. So the signal here is in white, right? The bright signal in the nucleus is in white. Whereas here, we see cells and we still see the nuclei, but they're not lit up. They're actually just pieces where the stain didn't go. So that means where the protein didn't go. And if you look at it, there's not really that much of a difference in intensity between what's in the nucleus versus what's sitting in the cytoplasm. But we know that the nucleus is a much smaller compartment than these areas of cytoplasm. So even though this stain doesn't really suggest it, the concentration of this protein in the nucleus must be pretty high. Because if it is not allowed to go into the nucleus, it stains the entire cytoplasm pretty brightly, right? So there's a ton of this protein in these cells. And the only difference here, based on a single amino acid change, is where it ends up. If it stays in the cytoplasm, or whether it's taken up and brought into the nucleus. And so you can see these are charged amino acids, and again, there's going to be slight changes in pH that are going to be helping these import signals to drive binding to the import receptors and then release in the nucleus. And we'll see how that goes in much more detail. But first, let's have you guys talk instead of me. So why do we need specific signals to direct proteins to the nucleus? And what types of proteins are we actually sending to the nucleus? So take a couple minutes and talk to each other, and I can walk around and answer questions if that's helpful. know how in the nucleus there's areas of active DNA and areas of inactive DNA, and those often are bound at different frequencies. So that's why you see the dark color. So this is within the nucleus. So in this area where it's all dark, you see that difference. Because there's not a nucleus but in this area. So yes, even within the nucleus, there's some areas where the DNA is so compact that you can't reach it. So that fluorescence will pop out. They're pretty the same intensity, right? Yeah. Thank you. All right. Why do we need specific signals to direct proteins to the nucleus? Why can't everything just occur by, I don't know, diffusion or of some kind? Who wants to offer an option? Are you waving to me or are you raising your hand? Yes.
We were just talking about generally how the cell needs signals because without it, different proteins would go in different locations or things would diffuse out that shouldn't diffuse and the cell would be as organized as it would with the signals.
Right? That's right. So he's saying without signals, the cell will get disorganized and stuff. They go places where it isn't needed or diffuse away from areas where it is needed. And so think about it, the nucleus isn't freely accessible to everything, right? We just talked about this. Those pores are fairly big, but they're not big enough to let everything just come in. So it's a separated area from the rest of the cytoplasm, so we need to have some way to tell some proteins that, okay, you are needed, you need to come in. So what are those proteins? What type of proteins interact with DNA? Yes? Transcription factors are a big thing, right? They are made and then go in, and they don't need to be there all the time. So they also need to come back out. So this process needs to be regulated. Is there anything else that needs to go into the nucleus? What happens if we go into mitosis? What do we do with our DNA? We split it, we duplicate it, we pull it apart, all that stuff needs to then be in the nucleus to do its job, right? So lots of proteins need to go into the nucleus. And then as I was walking around, there was a question about this picture. And they were saying, no, but isn't there like a dark nucleus in this one as well, right? Because this is not an even stain. And so these pictures are from the same kind of cell. So they would have the same size left and right. So what you don't see here, but that is present, is the cytoplasm of these cells that sits around them. And then here on the left, we see the signal sits in the nucleus, but it doesn't sit everywhere at the same intensity with the same frequency. And this is because not all DNA is equally accessible. Some DNA is much more tightly wound up, and so other proteins cannot go in and bind there. So we often see those nucleoli, those areas of a different intensity of DNA stain if you were to stain for the DNA. And if you stain for transcription factors, the really tightly compacted stuff that stains very brightly with a DNA stain doesn't really bind to transcription factors because they don't have access. The DNA is too compact. So that's why you see that difference in intensity. Let's see, what else? And then here in the cytoplasm, because it has no signal telling it to go somewhere special, it just kind of diffuses throughout intermediate, all the filaments, the other organelles. It just spreads out because there's nothing telling it to go someplace specific.